All right, so we will see what happens here as we go forth. All right, okay, thank you.
Well, good morning, class. Welcome to our science seminar for today. We've got lots of exciting science seminars this month of April. Today we'll be talking about soils. Next week we'll be talking about forestry. We've got pharmacology coming up. All kinds of exciting things in this last month of April for science seminars. So I hope you're enjoying it. Today we have a returning student, an alum from LeTourneau. Cara Case, who was Cara Harklerode way back then, long time ago. What year did you graduate?
2004.
2004, so a little bit more than 10 years ago. So there is life after LeTourneau. I know you guys have a hard time believing that. You just want to stay here forever. Armstrong and I decided to do. I mean, there's nothing else after LeTourneau, right? But Harklerode Case says that there is something, and she's going to tell us about the big world after LeTourneau. So I like to say that, since Cara works in soil, she has a lot of dirty stories to tell. But, that's never a good joke. You guys never like that. So let's not waste any more of her time. Let's welcome her here.
So I'm Cara Case, and I work at the Texas A&M Agri-Life Center in Overton, about 30 minutes away. And I'm going to talk to you about microbial roles in soil health and biogeocycling. And when I say I'm a soil scientist, people usually ask me, what is that? How did you even learn about that? Like, I didn't even know that was a thing. Well, it is. I love to garden. I've been gardening since I was about 12. And I enjoy getting muddy, as you can see in the picture. And when I came to LeTourneau as a student, I thought I would major in biology and then go on to graduate school at A&M and major in horticulture. There was a faculty member, a biology professor, Dr. Jarsper, here, and he was a fungi and soil expert. He loved all things soil, and when I took his ecology class, he taught us about all the complex things going on in soil and the soil ecosystem, and it was really just fascinating. And so the summer after my junior year, I took a soil science class from Stephen F. Austin University in Nacogdoches, near where my parents lived. And it was really a great application for all the physics and chemistry and biology that I had been learning at LeTourneau. So I was like, okay, this is what I want to go to graduate school for. And I'm just fascinated by how waste turns into fertile soil. It's really cool to me. And Jesus uses this as an analogy when he's talking to his disciples and telling them that he's going to have to die in order to live again and to bring them life and to bring us life. And he says, unless a grain of wheat falls into the earth and dies, it remains alone. But if it dies, it bears much fruit. So first, let's look at some of the soil regions across the globe. So these soil orders you can kind of think of as the biological kingdoms. So they're kind of the broad, highest classifications of soil across the globe. And how many of you are from the Midwest? Let me see your hands. Okay, several of you. And how many of you grew up on a farm? Yes, very good. So the Midwest, you can see, is this dark green mollisol, which develops under prairie vegetation. And what happens there is the grass roots grow really deep down, like sometimes 20 feet down, and they'll die at the end of the season. And then the next year, they grow deep down. And so you get this really deep organic soil that's really excellent for growing grain and corn and all those things. And that's why the Midwest is called the Corn Belt. And you can see there's really not that much of that dark green across the globe. There's a lot in Russia and Eastern Europe, some in China, some in Argentina, but that really fertile soil is not actually all that common. The light green soil is also pretty fertile, not as fertile as the mollisols. The yellow and the pink are probably the least fertile soils. These are developed usually under high humidity climates and a lot of heat, and it just leaches the nutrients out very quickly. Not that, like, most soils can become productive with good management, but some have a natural propensity for fertility better than other soils. And here's another map showing the agricultural area and the change in agricultural area. You can see that a lot of the, there it is, okay. So a lot of the developed countries have actually stabilized their agricultural area or actually slightly reduced it, whereas a lot of developing countries are still kind of expanding their agricultural area. And some of this expansion may be sustainable, probably a lot of it isn't. But the good news is globally, we've kind of stabilized our agricultural land area or even decreased it somewhat, while at the same time, even though the population of the world is growing, we're producing more food per person. And the reason, a big reason for that is because of recent advances in agriculture. Over the last 100 years or so, a lot of things have happened that really help us in our efficiency in agriculture. One of those things is industrial fixation of nitrogen. So during World War I, the Germans developed a process to convert atmospheric nitrogen into ammonia to use in their explosives for the war. And this is the Haber process. And then after the war, they were like, oh, hey, we can put this nitrogen on our crops. And it's much more efficient, like you can put a lot less ammonia than previously you had to put animal manures or something like that. They're bulky, heavy, and you have to put a lot more manure for the same amount of nitrogen. Another advance in agriculture is called the Green Revolution. This is a really cool story about Norman Borlaug here in the picture. He did research on wheat in Highland, Mexico, and he developed a short-stemmed, disease-resistant wheat that produced as much or more as the previous wheat varieties. But the previous wheat varieties would have problems because they were too tall and the wheat grains would get too heavy and they would fall over and it was really hard to harvest them. So, and then after he developed a variety from Mexico, he also developed varieties for India and Pakistan. And within 5 to 7 years, India and Pakistan went from being a net importer of wheat. They needed more wheat than they could produce to being a, a net exporter of wheat. So Norman Borlaug got the 1970 Nobel Peace Prize for his work in feeding millions of people. And then a third advance in agriculture is genetically modified crops. For example, herbicide-resistant corn and soybeans really help in allowing better weed control with fewer tractor passes on the fields. And this helps the plants because fewer tractor passes means less compaction in the field, and the roots can penetrate the soil deeper. And also it helps the farmer because they're paying for less diesel fuel for their tractors, less herbicide, less chemical, and also fewer man hours. But a lot of, so this has been used extensively, but a lot like the antibiotics in the medical field, like there is no silver bullet, and we've used it so much that now it's kind of become controversial, and like there are actually weeds that are developing resistance to this herbicide. So they'll spray herbicide and knock out most of the weeds, but then the pigweed that's resistant takes over. So we have to constantly be learning new things and learning new ways to produce food efficiently. So what do we do in Overton? There's kind of two divisions. There's the extension division that is kind of a public outreach service. So if you have questions about pest management, or you want to start a vegetable garden in your yard, or build a catfish pond on your property, or go into cattle or forestry enterprises, these are the people that you want to talk to and get information from. They're a great resource, and they have a lot of published material online that you can look at. On the research side, we kind of are focused more on, okay, how does the science of agriculture work? And we kind of generate scientific information to feed to the extension people who share it with the public. So, and in the research division, we have people specializing in cattle forage management, tropically adapted cattle, ornamental horticulture, legume and forage breeding, and also soil microbiology and soil health, which is the team that I'm working on. So what is soil health? There's a lot of hype lately about soil health, and what is it? So it's a continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. An older term for this is soil quality. It has chemical aspects. You want to balance your mineral nutrients. Physical aspects, your pore space, water holding capacity, how the water moves through your soil, and also biological, capturing solar energy and recycling that energy. And in addition to all these soil quality parameters, soil health kind of emphasizes the sustainability for the future and a closer look at biology. We have a lot of biological tools now, especially molecular biology, that we didn't have 20 or 30 years ago, and it's really enabled us to look closer at what's going on in the soil. So how do you build soil health? Minimize tillage or other disturbance, cover soil with plants or mulches, and this will protect the soil from the impact of raindrops and also prevent it from just baking in the sun. So it prevents a lot of daily temperature fluctuations and makes the soil kind of a more happy place for the soil microbes to be. Maintain vegetative growth throughout the year. The plant roots actually exude sugars and carbohydrates for the bacteria and fungi to eat, so that they will kind of attract those soil microbes to the rhizosphere, which is the soil right around the roots. And because the the bacteria and the fungi actually help bring mineral nutrients into the plant. So if you interrupt that plant, if you don't have plants growing on your soil for a time, then that interrupts the soil ecosystem and kind of breaks that chain. Also, add plant diversity through cover crops and crop rotation. Different plants from different plant families will exude slightly different chemicals or signaling molecules, and some fungi are specifically symbiotic with those plants or a different family of plants. So the more plant families you have in a given patch of soil, the more robust your system is, and if you have a disturbance or something, then it can recover faster. And also a balance of soil nutrients. If your chemistry is not balanced, if you have a deficiency in something or an excess, things aren't going to grow well. So the questions we're asking in our research is, what factors play the biggest role in soil health, and by how much? And how do marginal soils respond to soil health building practices? So this is kind of a diagram of the soil food web, kind of like the food chain, except there's a lot of things that interweave. So, for example, these arthropods up here, they eat nematodes, they eat fungi, they eat bacteria, they shred the organic matter. So all these characters have a lot of different functions, and if you disrupt one part, the rest kind of can compensate, but there's still a hole there. So, and here's the soil carbon cycle, kind of a simplified version. So the plants and the dead plant material and also animal wastes come and accumulate in this active soil organic matter fraction. And and this is kind of your fresh wastes that get broken down the fastest. The active fraction gets broken down probably within a growing season or two. The slow soil organic matter breaks down over, say, 10 or 20 years, whereas the passive soil organic matter is actually chemically bound to the soil aggregates, and it has a turnover time of hundreds of years. And so the soil microbes and fauna will chew up all that stuff and produce a lot of CO2 just by respiration. They also produce a little bit of methane, especially the methanogens that I'm going to talk about in a little bit. And here's the nitrogen cycle. The nitrogen cycle is closely tied to the carbon cycle just because the nitrogen is often bound up in that soil organic matter. One thing that we're focusing on is this denitrification here to atmospheric nitrogen and nitrous oxide. So atmospheric nitrogen is not a greenhouse gas, whereas nitrous oxide is kind of a potent greenhouse gas. So we're looking at what bacteria cause the denitrification to go towards atmospheric nitrogen versus nitrous oxide. So how do we take our samples? We use a hydraulic soil probe. You can see it on the back of this tractor that I'm driving here. And we pull soil cores 24 inches deep. And for DNA and chemistry analysis, we use a soil core with a diameter of 1.5 inches. But if we're doing root studies, we need more, so we take a 3-inch diameter core. We'll take those back to the lab, and then we cut them into depths. So the first 6 inches is our topsoil, and then there's usually a middle sandy region, and then the clay is usually 18 to 24 inches deep. So we'll take 2 or 3 of those cores, the depths from 2 or 3 of those cores, and mix those together for one sample. And this really helps create a representative sample, because when you're out in the field, sometimes there's a cow pat there and a gopher hole there, and if you don't pay attention to those things, then they're really going to mess up your results. So then we split it into subsamples and freeze some for DNA at -80 and chemistry at -20. So I really like this picture here because you can see all the different colors in the soil. And so like these 3 soils here they came from the same core. And this one is a little bit darker because of the organic material, and here's that middle sandy, kind of yellowish sand. And then your typical East Texas clay right there with the iron oxides. So we're air drying our samples here for pH and total carbon, total nitrogen analysis. And we'll also do a water extraction, which kind of tells us what is the active fraction of carbon, what is the active fraction of nitrogen, and that kind of represents the plant available nutrients. And then I'm also in charge of the ions and cations, the anions and cations, and I do that on ion chromatography. And so here I'm filtering for that ion chromatography. This is a clay sample. It's really thick there. So we have to get all those particulates out in order to run it on the IC. And then here's a sample chromatogram from the IC. So then we'll do DNA extraction in preparation for our biology work. We crush the frozen samples in a mortar and pestle until they're thawed. For clay samples, we do 3 cycles of freeze-thaw with liquid nitrogen. And liquid nitrogen has a boiling point of -196 Celsius or -321 Fahrenheit. So it's super cold, freezes that sample right away. We keep pounding on it until it thaws again, and then we put more liquid nitrogen. And so we do that 3 times to get it all crushed up and broken up. Then we'll shake them with garnet beads for 10 minutes, and that breaks up the actual cells and gets the DNA out. And then we'll use a spin column extraction kit that's specifically designed for soil samples. So before I go into what we do in our lab, I want to give you some background on polymerase chain reaction, or PCR. So this is kind of like a copy machine for DNA. And so we put our template in here, which is the green, and also the primers, which are these pink things here. So the primers determine what section of DNA you want to copy. And then the blue nucleotides are the building blocks, how you assemble the rest of the DNA and make the copies. So in the first step, we heat up the DNA to denature it, and so we're breaking all those hydrogen bonds between the 2 strands of DNA. Then in the second step, we lower the temperature enough for the primers to anneal to its complementary sequence. And then we raise the temperature slightly to the optimum temperature of the polymerase enzyme, which actually finishes the copy of that DNA segment. So at the end of the cycle, we'll have 2 copies of our template, where at first we just had 1. So then we'll do this for 40 cycles, each time doubling the number of DNA copies we had at the beginning of the cycle. So by the end, you have millions of copies of your DNA segment. So then we run it on a gel. We run it on a gel, and if you see these bright bands here, that means your reaction was successful, and that's a very good feeling. So now, for quantitative PCR, or qPCR, we use, the main difference is a fluorescent intercalating dye. And what that intercalating dye does, like this green bar here, is it gets between the rungs of the DNA molecule. And when it gets in between those rungs, it fluoresces a lot more than if it's just free floating in the reaction. So we can read that fluorescence and see how much double-stranded DNA we're accumulating over those cycles. So, and one nice thing about qPCR is you don't need a gel to read your results because it's showing you that fluorescence over your run. How many of you were in IEP and made a 3D printer? Oh yeah, oh yeah. Okay, so those stepper motors that you use in your 3D printer are also in this pipetting robot that we use to prepare our qPCR sample. And this is, I think this is really cool. I may have to get over here. So, I don't know if you can see it, but so it's picking up a pipette tip and then getting some sample and pipetting it into this 100-well ring that we do, that we run for all our samples. And this is just an example of, we scientists need you engineers. So go and be good engineers and help us poor science people. So then after that's done, we'll put it into this qPCR machine, and it will spin and read that fluorescence over each cycle. So the first thing we check after our qPCR is done is the melt curve analysis. And this is just kind of a double check to make sure that the target that you want to amplify or copy is actually the thing that you're copying. So all these multicolored lines here are our samples, and the red ones are our standards. And so those peaks are aligning pretty well. That's a good indication that we're amplifying what we want to amplify. So now we get to the quantitation part. So first, we find the CQ, or cycle of quantification, and that's the cycle at which the fluorescence of the sample exceeds the background fluorescence. So, so then we find where each of those standards, the red ones, cross over this line of quantification. Different softwares will call it different things, and finally there was some committee or something that said, okay, we're going to call it CQ. So wherever those standards cross over that line, so this one looks like it crosses over about 17 cycles. And so you can plot that with your calculated copies per standard, copies per reaction, and that gives you this linear regression here. And that's a shame you can't really see the writing. But, so that will give you a linear regression, and then you can plot where your unknowns come out on that graph. And we usually run 5 standards in triplicate, and those standards are an order of magnitude lower than the other. So we have anywhere from 100 copies to 1 million copies. We can find it. And oh yeah, we run our standards in triplicate just because there are so many unknowns going on in the qPCR reaction, and sometimes you can see that one of your standards is just off for whatever reason, or sometimes even two are off, but you still have that third one that you can use that gives you a good regression. So I'm going to talk to you a little bit about a few of our projects that we're working on. This methane cycling project is looking at low, medium, and high cattle stocking rates. So we have half of our pastures planted in clover, which does not get any nitrogen fertilizer because it's a legume. It fixes its own nitrogen, much like the Haber process that I talked about earlier. And the other half are planted in ryegrass. Ryegrass is not a legume, so it needs additional mineral fertilizer. And we're looking at methane production by methanogens and methane consumption by methanotrophs. So methanogens generate the methane, and then the methanotrophs use it as their energy source. And again, I apologize for the small writing. So these two prokaryote graphs kind of give us an indication of how many total bacteria are in the soil. And then these methanogen, the gene target for that is NCRA. And so this is, these graphs up here are stocking rate, and these are organic versus inorganic nitrogen source. So the organic is going to be the clover pastures, and the inorganic is going to be the ryegrass pastures. And then here we have the methanotroph, which the gene target for that is PMOA. So, and we kind of expect to find more bacteria in the high stocking rate, and also more bacteria in the organic clover pastures. And the reason for that is a lot of times inorganic fertilizers will, they're salts that you're putting on the field. And those salts can upset the isotonic solution or isotonic environment for the bacteria, and sometimes that interferes with their growth. So, and we kind of see what we expect to see. We can't really say statistically that some are higher just because we had so many outliers. This was our first sampling time point, and we've sampled twice after that, and we're hoping that our sampling method was a little better, and we'll get some good data out of that. Another project is a native nodulation project. So legumes, like I said before, form symbiotic relationships with bacteria that convert atmospheric nitrogen into usable form, those nitrogen fixers. And we're doing this experiment on very sandy, low fertility soils. And it's an intercropped system of sorghum and cowpea. So the cowpea is the legume that fixes the nitrogen, and then the sorghum is a grain. It's not a legume, but it's supposed to be really good in drought situations, and used as a livestock forage, and also for human food in some places. So some of our cowpeas are inoculated with the recommended rhizobia bacteria, and others we didn't inoculate, and we're testing to see if the native rhizobia in the soil will inoculate. How much will they inoculate? Is it better to inoculate, or is it better to leave the native rhizobia to inoculate? So far, we've counted numbers of nodules on the non-inoculated and the inoculated, and we've measured the mass of roots at different depths, but we haven't done a lot of analysis on this one yet. And also, we're doing a project involving Caddo Lake. And we're looking at mercury methylation. So mercury is mostly dry deposited into waterways, and then it's methylated by anaerobic bacteria. And this methyl mercury is actually a lot more bioavailable and a lot more toxic. And then it'll bioaccumulate in fish and other living tissues. So the questions we're asking are, what are the hotspots of methylating bacteria? Where do they hang out? And also, what role do invasive aquatic plants play in mercury cycling? So I really like this project because we got to go in canoes and do a bunch of sampling, and it was fun. So, and to close, if this presentation was really interesting to you and you'd be interested in working with us for the summer or even during the semester, there's a student worker position available, and the rate is $7.50 an hour for college students, or if you're graduating in May, you can also qualify as a student worker. Our position is about 75% lab work and 25% field work. It's about 30 minutes from Longview, and if you're interested, I have some applications over here as well as some brochures you can pick up. You can contact me at cara.case@ag.tamu.edu or my supervisor, anil.somanahali@ag.tamu.edu. And I've also heard just yesterday from Dr. Pemberton, the horticulture guy. He's looking for student workers too, and they would be working in the greenhouse and the field for kind of ornamental bedding plants kind of stuff. So with that, I'll thank you for your time and attention, and I'll take any questions.
So I would encourage you to look into those job opportunities, all you biology peoples out there. If you have more questions or Harper Road or States, take those now. So always be Harper Road to me. The Harper Road, unfortunately, was the case.
I took a lot of classes from Dr. DeBoer. A lot of classes.
I used to have to come to class with my safety goggles on because things would be thrown at me if students weren't happy.
I don't know anything about that.
All right.
Yes, sir.
You talked about inoculating plants. How do you do that?
You
I
take
mean,
the,
my mind is a hypodermic needle and sticking it in a root or something.
right. So we take the dry seed and we get it wet just a little bit, and then we sprinkle this black powder on it and stir it up, let them dry just for a day or so, and then we'll plant.
So does salvinia take up mercury? Is that what you're looking at?
That's kind of what we're looking at.
So does it?
Or do the anaerobic bacteria hang out on their roots? We don't know yet. We're still waiting on the results. So for the methyl mercury, apparently that's a really specialized analysis that you need expensive equipment for that we don't have, so we had to send those samples off to get analyzed. Yes?
So you used garnet
Yes.
to bust open the bacterial cell walls? I'm
Yes.
curious as to why that was used as opposed to maybe like sonification or something else.
I think it has rough edges. There is a bead beater that you can get that just like that, but I think the rough edges help to like break apart the cells, and we'll put the lysis buffer in there with them.
Would it seem to work better than the sonification?
Well, we haven't tried with sonification, but I imagine the soil particles would just eat away at your soil, at your sonification probe. That's a good question. Any others?
again. For all the chemistry people here, there's the American Chemical Society meeting tonight at 7 o'clock. Right? And Dr. Hathaway is in charge of that. There's a question for him. You can ask him. That's at 7 o'clock here. We're going to be having lunch with Cara over in the Joyce Room or the Corner Cafe. So if you want to learn more about summer opportunities from her, come and join us for lunch. And any other announcements we need to share before we go? No? Okay. Well, let's thank you.